Welcome back, everyone. Today we'll be studying about natural selection first. So, what is natural selection? Natural selection is the process in which the different organisms in a specific habitat compete with each other in order to obtain the nutrients present in it. Now, there are different types how natural selection works. For example, a predator has better vision to easily find its prey. Similarly, the prey can better camouflage with the environment in it to hide from the predator. Therefore, natural selection, we have to first realize that natural selection doesn't occur within a span of, for example, a few years. However, it occurs over thousands and thousands of years. Now, what is the difference of it with artificial selection? Artificial selection is the process in which humans specifically crossbreed only those organisms which are beneficial for them. For example, they help to increase the production of milk in cows or goats. This the advantages of this is that only those offspring are chosen which have the quality to show the most number of those characteristics. Similarly, now we'll be talking about the process of genetic engineering. What is genetic engineering? In genetic engineering, a particular gene is transferred from one cell to another. The most potent type of example of genetic engineering is insulin. In insulin, first, the gene for insulin is cleaved or taken out from a human chromosome. This is carried out by the process of restriction enzymes. Now, what are restriction enzymes? Restriction enzymes are just like scissors. You need to know that all these things written in bold are frequently asked in the exam. After removing the gene from the chromosome, it is inserted into the plasmid. However, before doing so, obviously, as the plasmid is sealed, it has to be cut by the same restriction enzymes. After inserting the gene into the plasmid, it is then resealed by the ligases. The ligases you can remember by the term glues. They're like glues, so they join them. Now after inserting the gene into the bacteria, of course, the bacteria replicates at an extremely rapid speed, and thousands of copies of the same thing are multiplied and replicated. Therefore, the human insulin, which is required for the normal human body, is obtained rapidly. Now, the plasmids in this case are referred to as vectors. Now, what are vectors? Vectors are anything which help to spread the thing. For example, the vector for dengue is Aedes mosquito. Now, the process of inserting the gene into the plasmid is referred to as splicing. Now, in this diagram, you can very well see how this process works. You can see that this was the whole bacterium and this was the ring of DNA. We broke this ring of DNA, and the human chromosome was cut from the nucleus of the human cell, and then after inserting it, it was resealed. Now, advantages of this form of insulin production are that firstly, no, no animals are hurt during the process. Secondly, a large amount of insulin can be harvested, and it is also a cheap process comparatively. The advantages of genetic engineering can't be limited to only insulin, and it can be used for a wide variety of purposes, such as curing another genetic diseases such as cystic fibrosis, and in the making of the enzyme renin, or to produce disease-resistant crops. However, for exam point of view, the most important example is insulin. Thank you for this listening to this video. I'll hope to see you again.